Hello and welcome to the presentation of our new paper in ICCV, titled Inverse 3D Microscopy Rendering for Embryo-Shaped Inference with Active Mesh. We are a biophysics group based out of Paris in France. The question that we are trying to tackle here broadly encapsulates the physical shape of embryos, which we define as a static problem. The reason behind this is because our data refers to 3D images of these embryos. Now, different ways to encapsulate the shapes of embryos would be 3D meshes, graphs with shape embeddings in the numerical format, or other 3D models. Now, before we talk about inverse rendering, let's understand what rendering means. As Wikipedia would define in the term of computer graphics, it is a process of generating a photorealistic or non-photorealistic image from input data such as 3D models. In our case, that would imply having a 3D mesh model of the embryo and generating an artificial image that would look like the same embryo. Therefore, inverse rendering would refer to the process of extracting or inferring such a 3D model that would be embedded in these real microscopy images. So, the way the paper tries to tackle this problem is by starting from a simple theory that the mesh model convolved by the point spread function gives us the microscopy image. Let me break it down. A point spread function is basically the point response of the microscope machine to each image that it takes. The microscopy image that is rendered is the data that we receive, or the data that biologists extract. Now, there are different models of meshes available. The one we are going to work with are triangular meshes. And we will assume that the point spread function will be a Gaussian one. In the proposed algorithm, both of these, the mesh and the point spread function, will be optimized simultaneously to achieve the perfect microscopy image. Now, as we know, a convolution operation in the spatial domain is the same as the respective signals Fourier transforms, but as a product, as we see on the screen. And doing this simple small translation would help us a lot by reducing the complexity of our problem from n to the power 6 to n log n cube, whereas n is assumed to be the discretized side of each image. However, that would require us to find an analytical formula of the Fourier transform of triangular meshes. There have been some computations and some proofs in this field from old papers, however, not one without any singularities that we could use the automatic differentiation tools like PyTorch or others. That is one of the main contributions of this paper, this proof of the Fourier transform for triangular meshes without singularities. The methodology of deltaMic involves, like I said, the cell shape inference and inferring the perfect point spread function such that their convolution yields an artificial image that is as close as possible to the 3D fluorescence microscopy image. For this, we use a weighted L2 norm as the loss. Some examples in the beginning that we tried to see if our method works were on simple shapes. So starting from a shell or a toroid to fit these shapes as you see on the screen. And they worked pretty accurately and quite fast. Then we moved on to our actual application of the work, as in to benchmark on young embryos from real microscopy data, We tried on across different species, as you can see, mice, ascidian embryos, and worms, C. elegans, and across different microscopy modalities, confocal and otherwise. We achieved pretty good results on these as well. I have attached some videos for the interest of the audience. These are the ascidian embryos, whereas these are the mice embryos, and you can see them, the shape of the initial mesh changing over time to fit the 3D image that is the ground truth in this problem. And to finish up, we have also performed benchmarking with an already existing method which used a combination of deep learning and energy minimization technique with a plugin in Fiji. However, our method beat their benchmark on their own data by at least one order of magnitude, as you can see. It is important to mention that their data is not embryo data, however, it is mouse organoids, which goes to show that our method is not just limited to application for embryos. Thank you.